So first of all, we will create for this workshop, we will create a folder. So I'm naming that folder as Autodoc. So I have created a folder called Autodoc. This is the first step. Then we'll download the target protein. Okay. So go to Google and type RCSB. So when you click it, it will open this page. And here you click RCSB. So this page opens. It is the protein data bank site. Our target protein, which we have read from the literature, is 1IEI. So this is the target protein. So we'll download this target protein. So you have to, there are different download options. First you have to download in the PDB format. Okay. So when you download it, make sure you keep that in the working folder. So we have created the working folder as Autodoc. So this is the, so first you create a folder, then you save the protein in the working folder. Okay, close this. So now we'll process this protein. There are some preliminary steps in this. For that, we will be using two softwares. One is UCS of Kimira. Okay. So when you open the UCS of Kimira, the interface will appear like this. So what you have to do is that you have to copy the path of the working folder. This is the working folder. So where we have kept the protein. So just click on the tab bar. So it will show the path. It is in the C drive in the desktop. Okay. Copy that path. Okay. And copy that path and go there and click on file and click on open. When you give open, it will show the folder. Then you paste that path. Then you give enter. When you paste that, it will show whatever is available in that folder in the right corner. So it is showing this folder. So now when you double click it, so it will show the protein. It will read that. It will show this protein. It is a ribbon model. Okay. So here, the basic steps what we have to know is, so here you go to the select. You can check the number of chains. So there is only one chain which is present. Then if you see the residues, it is having two set of residues. One is standard amino acids which are present in the protein, and the next is non-standard, that is heteroatoms which are present in the protein. So first what you should do, you have to select the water molecule. So if you select the water molecule, you can see the water molecule is present here. Okay. So now you go to actions, atoms and bonds, and you give delete. Then next is you go to select residue and then you click NAP. So if you click that, suppose nicotinamide adenine phosphate. So you can color that. I will show this. So this is nicotinamide adenine diphosphate. So go to actions, atoms and bonds, and you give delete. Okay. So now you are left with the protein and the ligand only. So now you go to select residue and ZES. So this is the target ligand. Okay. So suppose you can see this is residing in this place. So go to actions, atoms and bonds, and you give delete. So now you are left only with the protein. So I am going to save this file, save PDB, and you give naked protein, NAKED underscore. Don't leave any space in while using docking simulations. Naked protein. You can give dot PDB. So make sure there is one thing called use untransformed coordinates. It should be tick. Default it will be tick. If it is not there, you just click it. So it should not change any other coordinates. Then you give just enter. So it will save the naked protein. So you can check the folder. So we have a protein with other heteroatoms. We have only protein. Now we need to extract the ligand. Okay. Suppose in the naked ligand, if you see, there may be some missing residues which we have already seen. So go to tools, then you go for the structure editing. So in the structure editing, you give model refine loops. Okay, it will show this. Suppose these are the residues. So whenever you see this, what it will show whatever amino acids which are present. But if you see this area, first amino acid, there is no corresponding structure residue present. That is methionine. We have seen in the file. So if you want to build that, then what you have to do, you can create in the modeler, the account is free. You can create and you can model that missing residue using Kimira also. But you should have a valid license. So I'm clicking OK, but it connects to a server. If it is available, it will build it. Let us try it. So just give OK. Okay. So it is trying to connect to the server. Service Opal modeler, it has at present it is not available. Anyway, okay, we'll close this. We have saved the ligand. File, go to close session. So we have the naked protein with us. Again, I am going to open the original protein. So we'll repeat the same steps, but we are going to extract ligand this time. So go to select, that is residue, water, actions, atoms and bonds, delete. Then again I am going select residue, NAP, then action, atoms and bonds, delete. Then again go to select residue ZES. So this is ZES, but we need the ZES only. So go and click the select, invert selected models. You can give invert selected models. So it will go and select the protein instead of the ligand. So now go to action, atoms and bonds and give delete. So you are left with the ligand. So this is our ligand. So go file, save PDB and you give as ligand. So I am saving it as ligand.pdb and you give check there is use untransformed coordinates and then you give enter. So now you have saved the ligand. So you have the ligand, you have the naked protein. Okay. But we have not changed, we have not added the missing residue. So what we are going to do, we are going to use another tool. So file, go here, close session and you can close this. Okay. We are going to use another software called SBDB Viewer. Okay. So I will open that. So go there. 1 minute. I will open Swiss PDB Viewer. Yes. So the software name is Swiss PDB Viewer. So just click that exe file. When you click that exe file, it will give this pop-up. You close this pop-up, first pop-up. So now what you do, you just drag the naked protein to this area. You just drag this. When you drag this, it will give a pop-up. There are some amino acids which are missing. Your reconstruction of the whole side chain will be attempted. Affected amino acids will appear in pink. You just give OK. So then it will show whatever coordinates it has added and you close this. So here you can see there are some pink residues in it. Go to there are different tabs in this. Go to the wind and check control panel. If you see the control panel, you scroll down. So then you can see, yeah, missing residue is added. Okay. We have seen methionine, but some others are also missing. It is trying to add that. Okay. So that residue is added. So go to File, Save, Save current layer. So I am saving the current layer. So go to the desktop. Our target folder is Autodoc. We have created a folder name called Autodoc. So in that, you go there. So I am renaming the naked protein file name. So I am renaming this as underscore SBDB V. Okay. So I am giving the file as save. So our job is over. Missing residues are added. You can close these pop-ups. So we have preliminarily processed the protein. We have the ligand with us. So now we are ready to go to the Autodoc interface. So before going to Autodoc interface, the first ritual what you are going to do is that, so first go to my computer. So in my computer, you just go to C drive. So in the C drive, click program files into 86. In the program files into 86, you just go and then scroll down. You can see the Scripps Research Institute. So go there. There will be two folders, Autodoc and AutoDocVina. You double click Autodoc. If you see that, it will show the version. It is 4.2.6. You double click on 4.2.6 and then you can copy these two files. Copy and then we will place in the Autodoc folder. Okay, so this is the first ritual we are going to do before using Autodoc. So you can close this. So now go to Autodoc. So in the Autodoc, when you open that, it will take few seconds. Wait for a few seconds. It will open the interface like this. There are several tabs in this. We are going to use a few tabs. One is file. We'll be loading the protein, preparing the protein with the edit tab, and loading the ligand with the ligand tab and processing with the ligand tab. And then we'll be using grid tab for generating the grid box. Then we will be creating a program for docking. Then we will be running the Autodoc and AutoGrid. And after running the docking program, we'll be analyzing the results. So the first ritual what we are going to do is go to the file and click preferences. So now you set the preferences. So it will show many things. Just go to the startup directory. In the startup directory, you just remove one letter. So whatever is available, you remove one letter and click make default. Since we removed one letter, there is no such directory. It will ask for the working folder. So we have already created the folder in the desktop Autodoc. So click it, give make default and give set. Don't change any other things. Just make in the startup directory, what is your working folder? Select it, make default and set and close this. Okay. So this is the first step after opening the Autodoc. Then you go to file, read molecule. So we know we have processed this protein, naked protein, SBDBV. So open that. When you open that, it will be in stick model. Okay. It will be dull in appearance. You just present Control D from your keyboard. You can see the protein. Okay. So we have already known there are no hydrogens in it. Go to the edit tab. You can delete water from this tab also. But since water has been deleted from UCS of Kimira, I'm going to select hydrogen and go and add. In hydrogens, There are two types of hydrogens. One is polar hydrogen and one is non-polar hydrogen. Polar hydrogens are attached to oxygen, nitrogen, whereas the non-polar hydrogens are attached to the carbon atoms. They don't participate in any bonding. Okay. So, but by default, you select all hydrogens and click OK. Okay. So, it will add the hydrogens. You can see the white things. It is the hydrogen. Then go to edit hydrogens. And next is merge non-polar. I don't need non-polar hydrogens for my simulation. So, merge non-polar, you will have only polar hydrogens. Okay. Then go to edit tab. You give atoms, assign 84 type. That is Autodoc type. I'm changing these atoms to Autodoc type. Then edit, go to charges, compute, gas sticker. You can, or you can add Coleman charges also. Let us add Coleman charges. Okay, the total charges are added in this protein. You just give OK. Okay, then go to the ligand tab. Now the protein is ready. Then while preparing the grid box, I will discuss regarding that. Go to the ligand, give open, input open. So it by default it is in PDB-QT format. Just click this arrow, down arrow. You click all files. So now you know the co-crystallized ligand, that is ligand. You open that. Ligand is a small molecule. So as soon as it comes, the Autodoc adds the charges and the torsions or the flexibility in that molecule. So just give OK and then go to the ligand, output, save PDB-QT. So now you save it. So now we are ready with the ligand. Ligand preparation is very easy in the Autodoc. Okay. The next step is go to grid. So grid macromolecule you can choose. So here choose means the macromolecule. Macro means bigger. So naked protein, this. Select the molecule. Okay. So it should give this pop-up contains no non-bonded atoms. You just give OK. So now we are saving the protein in the PDB-QT format. As soon as it gets saved, the color will change to this. From pink, it will change to this color. Okay. Then go to grid, set map types. We know the proteins are having carbon, hydrogen, oxygen, sulfur, nitrogen, whereas the ligands are having different atoms. We know this ligand is having bromine, chlorine, fluorine, like that. So you click directly. So you can see here the map types are only for the proteins, not for the ligands. So I am going to change this as between C and HD you add bromine B or bromine, then you give chlorine, then you give fluorine. Okay, then give accept. So you have to give the map types for the ligands also. So then you go to macromolecule, the grid, set map types, choose ligand. We know the ligand is here. Select the ligand. Then you go to grid, grid box. So grid box is the area where the docking is going to happen. So this is the area. We don't need the entire docking. If you don't know the active site, you can give blind docking. But we know the active site where the ligand is already present. So these are wheels. So you can scroll this and change. I am giving a shortcut. Go there. These are the XYZ coordinates. You right click. It will open a pop-up. You just change to 60. You give apply. Okay. Then you change this 60. Apply. Okay. Then again you go there. Change it to 60. Apply. Okay. Whatever we are going to do is the redocking. Suppose if I have a molecule which is bigger than this ligand, that is why I am changing the grid size. Okay. You can note these coordinates you can use in the publication also. Suppose here you can give one more thing. In the center tab, you press center on the ligand. So now you can see that the ligand is not, it is inside this grid box. Suppose you want to check it. If you unclick the protein, you can see there is no ligand which is outside the box. Okay. So now you go to file, output, grid dimensions file. So grid dimensions file XYZ, if you want to note in a paper, you can note down afterwards also. You just give grid dot txt. Okay, save. Then file, close, saving current. So your grid is ready. So now you would go to grid. Out, there is no need to give other options. You go to save GPF. You just give grid dot GPF. There is a grid parameter file. You can save it. So we have processed the protein, we have processed the ligand, we have generated the grid parameter file. Then you go to docking. So here in the docking, you have to make the protein rigid. You may make flexibility in some residues if needed. So I am at present making as set rigid frame. We know the naked underscore protein SBDBV PDBQT is the protein with torsions and charges. So I am selecting it. Then you go to docking, ligand, choose. So ligand is this, select the ligand. So now you can see in the map types, bromine, chlorine, fluorine are added. So now you give accept. Then you go to docking, then search parameters and you select genetic algorithm. So here it will show a pop-up. There are different things. No need to change this at present. Number of GA runs can be increased to 60 to get better results. Or you may see the number of evals. There are three options: medium, short and long. By default it is in medium. I am giving as it is. Give accept. Then go to docking. Then output Lamarckian. So you give doc dot dpf. Okay, this is a docking parameter file. So we just save it. So we have the grid parameter file, we have the doc parameter file. Now we'll run the simulation. Run auto grid. First you have to give the program path name. We already saved the execution file. So go to browse autogrid.exe. We know the GPF file program, that is a parameter file name. Parameter file name is grid.gpf. The output will be grid.glg. You just press launch. So you can see in the folder the all the simulations will run. It will generate the map files. Previously we don't have this. So you can see the map files are generated for every atom. Okay. The interaction between the protein and the ligand will be calculated as a simulation. So when this box pop-up closes, the grid will be generated. So it has closed. We'll go there. So in the Autodoc, we'll see you have the grid.glg. So any file you can open with Notepad or WordPad. I am opening with Notepad. So you can see, so it has generated a program file. So you can see the, here it has many calculations. Okay. So it will have the grid box. So it will show as successful completion. Here you can see successful completion at the bottom. So there you can see the grid box also, XYZ dimensions. It will show in this. So here you can see the how it has calculated, how the energies are calculated. So you can close this. Okay. Now we will run the Autodoc. So run Autodoc. So you go to program, path name, that is autodoc.exe, give it. Then the parameter file name is doc.dpf, open. So it will generate a log file. You just give launch. So this will take few minutes. As soon as it gets completed, it will generate a doc log file. So where it will show the 10 poses of the ligand, and how it has interacted at the receptor site. So that could be read by analysis. The binding energies, if for every atom, it will put a cluster, it will put a calculation, and it will give the total energy. Then you can see the interacting atoms from this. You can note down, but you cannot visualize the interacting atoms from this. So for that, we will be using Biovia Discovery Studio. Okay, or LickPlot can be used. So basically, so in the Autodoc, we have used different things. So one is file, set preferences. Then you go to the folder which you are working. Then in the edit tab, you can have the processing the protein, that is adding the charges, adding the hydrogens. Then you can delete water also. Then assign AD4 type. Then you will go to the ligand tab where you will give the input for the ligand, and then you will save the ligand in the PDB Qt format. When you go to the grid tab, you will be processing the grid box, setting the grid box and setting the map types, and you will be saving the grid parameter file. So in the docking, you will be creating a doc parameter file. You will use the genetic algorithms, and you will be creating number of poses. That is, I have given 10 poses. So you will create the doc parameter file. Then in the run tab, you will be running the simulation. You will be creating the the grid log file, and you will be generating the doc log file. So after that, we will be seeing the results in the analysis tabulation. Since it depends upon the speed of your computer, my computer is i3, so it is taking some time. If your version is i7, or if you have the higher version, if you work in the GPU, it will be within few seconds this program will run. Otherwise, it will take some time. Okay, so this is the basic of docking. We'll wait for a few minutes such that this pop-up will close. Then you can go to the analysis tab where you will be loading the DLG file. If you load the DLG file, it will show the uh, you can uh, you can rank the ligands based on their least energy. So the least energy will be placed at the top, and the highest energy will be placed at the bottom, and the interacting residues will be shown. So I already discussed in the basic file that what are the interacting. There are 10 interacting amino acid residues, out of which 3 are having the hydrogen bonding. We'll check that here, but we cannot check the distance of the hydrogen bonding. That could be visualized with LickPlot. While analyzing, I will be discussing that. So it is a very useful tool. When you want to run another ligand, you have to go with the same steps. You have to create the grid box. In this case, since we removed the ligand and again we are trying to simulate it, it is going and placing inside that place where it is residing. It was residing. But when you use another ligand, it is not the place for that. It will not show that. It may be outside the protein also. But if you set the grid box XYZ dimensions, the ligand will be simulated in that space only. I hope you understand this concept. Okay. So that is why we have increased the grid size, which you can accommodate a bigger ligand in the space. So I hope it will give an outline of this up to uh, Autodoc run. Okay. So we'll wait since it is taking time. Uh, okay. So I hope it will take another 2 or 3 minutes. So we have used suppose if we see the Autodoc folder. So previously we had only few files. Now you can see how many files it has run. It has run many files. So now if you want to know the program has completed or not, if you see the doc DLG, it will be 0 KB. then it shows it is running. Suppose if you see the grid GLG, it is creating some KB. So when the program gets completed, it will show as some KB. So we'll see the program. No, it has not completed. We'll wait for another few minutes. Okay. So we'll check this. Okay. So since the program is running for few minutes, I hope you will wait. Other tabs I have not used. If you get familiarized with it, you can run Autodoc Vina in the same. You can analyze the Autodoc Vina also, where you will be using multiple ligands. In the next part of the video, we'll use pyrx, and we'll be using easydockvina in the third part. Okay, the first part will go for single ligand docking with this. Okay, so since it is taking time, so let us wait. How will we analyze this results? Yes, it has completed. So now we will go to edit, delete all molecules. So there should not be any molecule in this interface. So I will show how the log file appears. So now you can see it is 77 KB. So you can copy this. So copy this and paste. So now you can change this to text file. Then I am keeping the original file and creating a text file, txt. Okay. So if you change the extension, don't worry. You just give, it's a copy file only. So now you see the text file. So it will be like this. So when you scroll down the bottom, it has 10 poses with the binding energies. It has put lot of calculations. It will show the Ki kilocal per mole, whatever energy levels, all these things will be reflected in this. So here also you will get successful completion. Okay. So now we will analyze this. Okay. So go to, since already the folder preference has been given, go to analyze, dockings, open. So there is only one log file. You go to doc log file. You open. So read 10 docked confirmations from the doc TLG. You give just OK. Then you need a protein to study the interactions. So analyze, macromolecule, open. So it will automatically open the protein. So here is the ligand. Then go to analyze, confirmations, play ranked by energy. It will show a pop-up. You just click the AND button. So it will show this pop-up. So then you tick build hydrogen bonds. So the first pose. So this is the pose number. This is the hydrogen. So it is having an interaction with threonine 19, tryptophan 20. We know tryptophan 20 is already having a hydrogen bond in the when it is submitted. So I will go for, we know tryptophan 20, histidine 110 and tryptophan 111 where we have been already submitted. So we will check this. Go to the next pose. So it is the same thing. Go to the next pose. It is also same thing. Threonine 19, tryptophan 20. Go to next. There are only two hydrogen bonds. The same thing. Go to fifth pose. It is having only one hydrogen bonding. Tryptophan 20. Then the same thing. Threonine 19, tryptophan 20. Then seventh pose. You can see here we have got two hydrogen bonds out of three. Tyrosine 48 and histidine 110. So not only based on energy, but based on the hydrogen bonding also you have to select. So let us see whether we get three bonds. We will go check for up to 10 poses. You remember the seventh pose. Then you go to eighth. It is also same. Okay. Then 10th pose. So this is the 10th pose. Okay. So what we will do, we'll go to the 7th pose. So we are having two hydrogen bonds and the binding energy, you can see show info. So the binding energy is minus 10.07. So we will restrict to this and you just give right complex. Okay. So this will have this. You have to save in PDB file. So you have to click all. Okay. And then your file name should be complex dot pdb. Okay. So just save it. Okay. Then you can close these pop-ups. Okay. So we have the PDB file complex.pdb. So how will you analyze this? We know the energy level is minus 10.07. You just open with Biovia Discovery Studio. Since it has been already installed, I am giving open with Biovia Discovery Studio. So when you open that, it will show this. So here you have to use the right and left keys so you can rotate it. So you have the ligand and the protein. Okay. So here what you do, go to scripts and you click ligand interactions. Show ligand binding site atoms. So it will show this. So what are the hydrogen bonds? What are the, so you can rotate this. What are the hydrogen bonds present? Okay. So here what you do, so you have to represent properly while you are going for publication. So the molecule should be visible. Okay. Suppose there are any other additional ligands in this, you just click and delete. Okay. So now you go to right click that and then you give labels. Then add labels. So what I am going to do, I am adding the amino acids as labels and color as black. Okay. And you give apply and okay. So the amino acid residue, since it is a black background, you are not able to see it. Okay. Then go to scripts, visualization, go to publication quality. So you can use this for publication. Since the ligand atoms are in the same color, I'm going to rename it. Color, change the color. So you can color by element. So you can see sulfur, oxygen and nitrogen is in blue. So you can see the ligand. So this you can go for, this you can save it. So you can save this and you can show the 2D diagram also. If you click the 2D diagram at the left side, so you can see two hydrogen bonds are there and what are the bonds, different types of bonds, conventional bonds, pi-pi stacked bonds, pi-alkyl bonds, like that. If you get a red bond, it is an unfavorable bond. So I want to save this file and use it for my publication. You click here, it is a 3D image, file, save as, then you give image file. Okay. So I am going to save as complex shift underscore 3D. Okay. So it's a PNG image. You can save in JPEG also. So again, you click here. It will show the pixels. You give OK. Then you click here, file, save as. So I am giving it as 2D. 2D. Then you give save. So you can, this will also show the pixels. So give OK. Then you close it. So you can see the 2D diagram and 3D diagram. You can show the image. You can use it for the publication. Okay. Okay. So this is the 2D diagram. You can save it in the 3D format also. You can select properly and then you can save it. So it's that's all about the Autodoc. You are ready with, you have docked and you are ready with the results. Okay. So suppose if you want to open the file in LickPlot, so you can go and open with LickPlot also, okay, to see the hydrogen bond distances. So I that is also free software. You can get the permission. and then you can use the LickPlot. Okay, see here you can go to the softwares and then go to it is Lick plus new. Every year that gets renewed. It's a jar file. You open the jar file. Like it will appear like this. So go to file, open the open PDB file. You give just a PDB file. Go to browse. So our folder is in the desktop in the Autodoc. So it is the Autodoc. So that is the complex.pdb file. You just give it. So it is ZES unknown. Just give run. So it gives the plot and it gives the distance. So you can see the distance of the hydrogen bonding. How much is the hydrogen bonding distance? So even if this is 298 also, it is showing the hydrogen bond. You have got 3 hydrogen bonds. Where in the Discovery Studio, you got only 2 bonds. So it depends which visualizer you are going to use. So you can use this. You can copy this image and paste in the, you can save it in the PDF, or you can copy this and put in the image file also. Okay. So if you enlarge this, you can have a clarity of this. You go to file and you can save. So suppose you can save in the PDF file. Suppose you can copy and paste in the image file also. Okay. So it's a basic outline of the Autodoc. I hope you would have understood this. In the next part, we'll go for multiple ligands. Thank you.